All right, welcome everybody to our VacCAP webinar series. My name is Amaya Tucha and I'm part of the extension team for the VacCAP grant. Today we will be discussing two new throughput genotyping platforms for blueberry and cranberry with Dr. Nala Basile and Dr. Sean Clare. Dr. Nala Basile has served as the plant geneticist at the National Clonal Germplasm Repository in Corvallis, Oregon since 2002. Her primary research focus has been molecular marker development and applications to germplasm management and diversity evaluation in plants, with an emphasis on temperate fruits, nuts, mint, and hop. She led the genotyping team of Rose Breed in developing the first arrays for apple, peach, cherry, and strawberry. She is currently leading the genotyping team in the VacCAP project with the objective of developing high-throughput genotyping platform for blueberries and cranberries. And Dr. Sean Clare is currently a postdoctoral researcher working under Dr. Nala Basile since July of 2022. His work focuses on assessing genotyping platforms, mapping traits, and developing breeder-friendly markers that can be transferred to the community. Previously, he spent the past 7 years working on mapping and validating disease resistance genes in barley. So we are very excited to have both of these wonderful speakers today for this webinar. I just want to remind everybody before we start to keep yourself muted. If you have any questions, please put them in the chat box and we will ask those questions to the speaker at the end during the discussion period. And you can also put some of those questions on the Q&A. And so with that introduction, I'll just let the speakers take it over.
Thank you very much, Amaya, for the introduction. And thank you all for joining us today. So as Amaya said, Sean and I will be describing to you two new Flex-Seq EX-L high-throughput genotyping platforms for blueberry and cranberry. Oops. Okay, not working. Okay, so I'll give a very brief overview of the VacCAP and the Pangenome and SNP catalogs. Then Sean will pick up on the data formats that are expected from these platforms and the results from screening a cranberry diversity set in the testing phase with the cranberry platform, and then one for the blueberry platform with the blueberry diversity set. So if some of you do not know, the VacCAP is an SCRI funded project that is led by Massimo Iorizzo. And it's a multi-institutional project that has more than 50 partners and 25 institutions across multiple countries, including the US. There are 5 main objectives in the VacCAP, the first of which is developing genomic resources that include a pangenome, SNP catalogs, and a genotyping platform. So I'll be describing today. And the genotyping platform will be used for downstream genetic mapping for fruit quality traits. It will inform future assays for some of these quality traits, and that we have an objective of which quality traits we want to prioritize, and of course, transfer knowledge of everything we're developing and learning to our stakeholders and the industries. Oops. So starting with the pangenome, the blueberry and the cranberry communities using molecular diversity data as well as pedigree information selected 12 northern high-bush blueberry representative of the diversity of northern high-bush blueberry and 12 southern high-bush blueberry accessions that are representative of the diversity of southern high-bush. and 12 Cranberry accessions. These were sequenced individually, and this work, the pangenome work, was done by Patrick Edger at Michigan State University and Alan Yoka, and I believe that there is a webinar about it, and Amaya can put the link to the webinar if anybody's interested in the details. But I'm only going to give a very brief overview of what was done. So once the sequencing of these accessions happened, they were annotated. Then progressive cactus whole genome alignment was used to construct genome graphs to identify genomic variants across blueberry and cranberry and to estimate ancestral states. Then OrthoFinder 2 was used to identify orthogenes, paralogs, and compute comparative genomic statistics. And then SynMap was used to identify the synthetic cores. In the graph on the bottom left, you see on the x-axis the number of genomes of cranberry, and on the y-axis the number of genes. So I know the writing is very, very small, but the only objective of this is to show you in the pink graph, you see as the number of genomes, cranberry genomes sequenced in cranberry at around 11 genomes, the number of core genes remain around 15,000. So that tells us that we have gotten to most of the core genes in cranberry. In the gray graph, you see that the number of accessory genes is continuing to increase even at 12 cranberry genomes. So that tells us that if we want to get all of the accessory genes in cranberry, we need to do more sequencing. Now, the three graphs on the bottom, the first one is the number of cranberry core genes, which is around 15,000 in each of the accessions. So that's the panel in pink. And then in gray below is the number of accessory genes, which ranges between different accessions in cranberry. It goes between 10,000 and 16,000 genes, while in blueberry, in the northern high bush and the southern high bush, we have about 20,000 core genes, and the number of accessory genes is stable among accessions, and it's around 12,500. So that's a brief overview of the pangenome, but what it did is it allowed us to identify core and accessory genes for each of these crops to target. So then we developed a SNP catalog. The same pipeline was used for de novo SNP detection, both in cranberry and blueberry. Dr. Ping Zhang, she's at Washington State University, developed the cranberry SNP catalog. It included the de novo SNPs identified from the 12 cranberry accessions that were sequenced. And we added previously identified SNPs, 8 of them, that were identified by association mapping from the Zalapa group, and they provided that information to Ping. For the SNP catalog, core genes and accessory genes and trait-associated SNPs were prioritized. Now, the blueberry SNP catalog included more sequences. Mandy Driscoll, who's now at Fall Creek, she did the SNP calling. So the catalog included de novo SNPs identified from 47 diverse accessions. So those were the 24 that we have sequenced, as well as 23 provided to us by the Breeding Insight Project. We also included SNPs from transcriptome sequences of 16 cultivars that were not part of the diversity set that was sequenced, that we sequenced or breeding insight sequenced. And we had a lot of community members that provided previously associated SNPs, 13 studies from 8 association mapping studies and 5 biparental maps. So all of those made up the blueberry SNP catalog. And again, core, accessory genes, and trait-associated SNPs were prioritized. Now, for the genotyping platform, we wanted the platform to interrogate the targeted SNPs we wanted. We wanted a high recovery of those targeted SNPs. We wanted accurate genotyping. We wanted the platform to be flexible, and we wanted reproducible genotype calling. Of course, we wanted cost effectiveness, so that we all need cost effectiveness. And it was very important to us that it's a platform that can generate dosage in the tetraploid cultivated blueberry. So the community agreed to choose, we chose the Flex-Seq EX-L for multiple reasons. It's an approach that is hybridization-based capture. It uses 2 probes. It generates 200 to 300 base pair sequences. So that would include the targeted variant as well as other variants in that region. And it is easy. All we have to do is collect leaf samples, and Rapid Genomics, LGC, does the DNA extraction as well as the genotyping, and we just get the data back. So we also formed the VacCAP consortium to allow us to get cost effectiveness from the large number of orders in 1 year. And if anybody's interested in joining the consortium, please contact me. So for the testing set, the diversity set that we used for testing the platform, in cranberries, it consisted mostly of the American cranberry diploid samples, so Vaccinium macrocarpon, but it also included 3 samples of Vaccinium oxycoccus, the common cranberry, and 5 hybrid samples. In blueberries, we compiled samples from a large number of community members, and the diversity set included 72 northern highbush blueberry samples that are tetraploid, 72 that are southern highbush blueberry, also tetraploid. We also wanted to see how it performs in rabbit eye, that is hexaploid, so we included 21 samples there. And then we had 27 samples from other species. Some are founders or progenitors to the corymbosum or northern highbush and southern highbush blueberries, and some are of interest to introgress into breedings or in active breeding programs. So we had 192 sample diversity set of cranberry and 192 diversity set of blueberries. So a subset was used to develop a 50K loci, and then out of that, the best performing loci were determined and generated for the production, for doing the genotyping downstream. So in cranberry, we had 17,500 loci, and then in blueberries, we ended up with 22,000 loci. And next, Sean will continue the rest of the talk, and we'll give you now, we'll talk to you about the data formats that we get back from rapid genomics after the genotyping.
Yes. So Rapid will give you up to 3 different files back. The first will be this FASTQ file, VCF files, or their custom haplotype files. So this FASTQ file is very similar to a FASTA file. It's your raw sequencing reads that has built-in quality scores. So at the top line, you have starts with a @ symbol, and it has a lot of metadata about that read. You'll then have your sequencing read, this plus sign, and then your 4th line is the quality scores for every single nucleotide along that read. And you can look up how that scale works online if you're really interested in it. So the variant call format, this is all your variants for all your samples. And so the top, you have a lot of lines that start with 2 hashes. This will be a lot of different information about what acronyms mean within this file and lots of other metadata. You then have a single header line. This will have starts with 1 hash. So then you have your chromosome position, ID, the reference calls, and then all your sample names. And then from there, you get this. This is the start of the next line. So this includes a probe name, position, the reference, and alternate allele. So this one is actually, you can't see my mouse, but so it has an A as the reference and GT as the alternative alleles. You then have a quality score, and then lots and lots of metadata about this single probe and all its samples. And then here's the actual data. So here we have the tetraploid blueberries, and so where you see the 000 with slashes in between them, or 00/1/1, that would be heterozygous for this probe. And then after that, you get colons, which will be other metadata about that. So the allelic depth, total depth, other quality scores that you can refer back to at the top of the VCF. And then the last file is this haplotype file. And this is combining all of your aligned reads. So this is processed FASTQ files into BAM files, and then using that VCF file to assemble haplotypes for each probe. So this file format's much simpler, includes your probe ID, and then it will list every single variant from the VCF for that probe, and then has a list of all of the genomic positions within that, whatever reference you align to. And then after that, you'll get long haplotype nucleotide sequences at the end of that. You then have a colon, which will then provide the read depth for that haplotype. So for this one, it has on the first haplotype a read depth of 9. And then the ones in the middle have much lower read coverage, and then the last one has a read depth of 11. So if it's a really low read depth, it probably is just a sequencing error. Okay. So before we get into the results, this is all very preliminary results, and it's also what data you can extract from these. This platform is going to be dependent on how diverse your accessions are and how much sequencing depth you request or consider accurate for whether you're just doing detection or dosage calling. So for the Cranberry set, this is all called as diploid, and then I filtered the data for biallelic SNPs quality scores of above 30, mapping quality scores above 30, and read depth of 6, and missing data for at 10%. So if a variant had more than 10% of samples missing data, that variant was removed. So here is a heat plot across the genome of whether a locus was recovered. And so if it's green, we have a recovered locus. If it's red, it's unrecovered after my filtering. So the initial design had the loci spaced 27 kb apart. And before many filtering, we have essentially 100% recovery on these samples. And then after my refiltering, the spacing between those drops to around 32 kb, which is still very good. So this is, if we look at that on a per sample basis, average number of sites on these within each sample is around 400,000. And the mean depth is between 50x and 100x. And then after filtering, we have around 60,000 variants with a depth between 50 and 175x. And so to put numbers back to that first figure, we're only losing three probes are unrecovered. And then after filtering, we're getting around 12,000 loci back, which is around 72%. And right now we're looking at why there's such a large drop, whether it's a few samples skewing our results, or if these loci are so diverse and multi-allelic that my filtering steps are removing them since I filtered for biallelic SNPs. If we split that into all the SNP classes that Nala mentioned earlier, we get uniform recovery at each type of class, so we're not having any issues with recovery based on the SNP class. We look at chromosomes, so here we have pre-filtering on the left and post-filtering on the right. All chromosomes have uniform recovery across all loci, so there's no issues with chromosomes. And then this is the number of variants per locus. And so on the left, we have pre-filtering, on the right, post-filtering. And before any filtering, we have 29 different loci on average for each locus. And then after filtering, this drops to around 5. And then part of that's because I'm only filtering for biallelic SNPs that most people would prefer to use. If we split that on the chromosome level, that rough average of 5 holds true across all chromosomes. So here we have chromosome 1 all the way across to chromosome 12 of Cranberry. And so into Blueberry, this was called, fillers were very similar, apart from depth was increased to 10 reads to ensure that we had grid sampled every single genome of tetraploid blueberry. And then we also split those other blueberries into Cyanococcus and non-Cyanococcus samples. So again, this is the heat map of recovery across the genome. At the top, we have pre-filtering, and then we go from northern highbush, southern highbush, rabbit eye, Cyanococcus, and non-Cyanococcus. And so the spacing on this design is 31 kb apart, and then pre-filtering, we have very good recovery, over 95%. In northern, southern highbush and rabbit eye, we're still getting above 90% recovery, and the spacing's 35 kb apart. On Cyanococcus, I believe we had around 75% recovery with 43 kb spacing, and then non-Cyanococcus, I think it was around 60% recovery. I could get the numbers wrong. And then average spacing is 54 kb. So it's still much more dense than average LD in Blueberry, which is 70 kb. So again, looking at on a sample level, here these are color coded based on the class. And here we have average read depth again between 50 and 100 x, and then but this time we have 3.75 million variants. Post filtering, we're getting over 400,000 in the cultivated types, so northern and southern highbush and your rabbit eye. The Cyanococcus dropping to around 350,000, and then non-Cyanococcus just above 275,000 variants. And then average read depth again is between 50 and 150x. So to make sure this data was behaving as we believed it should, we took a subsample of 2,000 SNPs and constructed this phylogenetic tree. And so this phylogenetic tree is rooted by the multicolored clade labeled non-Cyanococcus, and then we move into the like beigey yellow color with Cyanococcus samples, and then rabbit eye in purple. And then we have some mixed and wild accessions for core and balsam, so hybrids between those wilds and northern high bushes in green. And then we have 2 large clades for northern and southern high bush. To put numbers back to that first density plot, here we have, before filtering, we recovered 21,000 loci, which is at over 95%, and then still getting close to 20,000 loci in the cultivated northern high bush, southern high bush, and rabbit eye. And then Cyanococcus, we're still getting over 16,000 loci, and non-Cyanococcus over 13,000 loci recovered. If we look at the SNP class again, we're getting uniform recovery across all those classes, so there's no issues of any SNP class. And then again on chromosome, we have the pre-filtering on the left and then going through to non-Cyanococcus on the right. We get uniform recovery on all chromosomes. And there's, on your northern high bush, you're getting over 1,500 loci recovered. And then on non-Cyanococcus, you're still getting close to 1,000 loci recovered per chromosome. If we look at the number of variants per locus, before filtering, this is at 181 different variants on average. And then this drops to between 10 and 7 across the different classes we have. So northern high bush has slightly more, and then we drop down to just below 8 on non-Cyanococcus samples. If we split that out again on chromosome, the trend stays true across each class. So northern high bush at the top there stays around 10, and then non-Cyanococcus is dropping down to 7 and 8. We did have some issue with some of the samples, which go back to the Cranberry, and we're still looking at this. They were skewing the recovery data so much that we decided to remove them from the data. And then here we are looking at them on a by sample case. And so this middle block here that's all colored with the hash marks, this is the number of loci recovered on that sample. And then on the right, we have the number of variants called within the loci recovered. And so you can see most of these samples are ranging from 350 to 450 variants called within these loci. But the ones in red and have the asterisks on the right, these were the ones that we had removed from the data. And they have significantly less variants at 200 to 250,000 variants. And that was directly correlated to being a poor quality sample that Rapid Genomics provided back to us. And so in conclusion, this Flex-Seq platform has excellent recovery on blueberry and cranberry, including those distant related accessions. There's uniform recovery across the genome and SNP classes. And then if you do want accurate dosage calls, you'll need higher sequencing depth. I'd like to acknowledge all our partners in this and any individuals that have been helping us along the way. Any questions?
We are happy to take questions.
Yeah. I don't see any questions on the chat, but if people have questions, if you just want to, you can just raise your hand, and if you want to ask your question, and if you don't want to do it that way, please go ahead and type it on the chat, and I'll be happy to read it out to the speakers. Seems like everybody's very shy or very quiet today.
Or everything was so clear.
The perfect presentation.
If you have additional questions that come up later after the presentation, you can email Sean and I with questions. We're always happy to help.
I have a question.
Yes.
This is Jennifer.
Yes, Jennifer.
You labeled something common cranberry. I never heard that before. You said it was
The
Vaccinium
Vaccinium oxycoccus.
Yeah.
Oh, you didn't hear that before?
No.
Okay, so what do you think we should call it, Jennifer?
Well, I don't think of
Or
it as common.
oxycoccus, the common name. We were trying to use just common name.
We usually just call it small fruited cranberry.
Small fruited. Okay, we can do small fruited.
Yeah, as opposed to macrocarpon, which is large fruited cranberry.
Okay, we can do that. Thank you very much for telling us. Small fruited cranberry.
This is Rod. Have you guys heard of mountain cranberry? Has it been called that too? But I do like the small fruited probably better anyway.
Okay, I like the small fruited. Okay, we can do small fruited.
Yep.
Cranberry. Thank you.
Yep.
Since I'm on the microphone and I'm probably the least educated in this of this group, could you go to the 60,000 foot level and tell me, I guess, does this work just help to eventually identify genes, or could you give me a layperson, really quick layperson?
Yeah, Rod, yeah, I can, because there are, let's say, 60,000 variants in cranberry. So there is a very good dense coverage of the genome. So if you have if you do, let's say you're interested in a phenotype, whatever phenotype it is, you do the phenotyping for root rot, let's say, okay? So then when you genotype the same individuals that you phenotype, because there is a dense coverage of the genome with these markers, you are likely to identify the region that controls that trait, and then eventually develop markers that can diagnose that trait. Did that make sense to you, Rod?
Yep, that was very clear. Thank you. That was good.
You're very welcome.
Okay, we have Hamid that has his hand
I have
raised.
one question.
Let's give Hamid his opportunity to ask his question. He's been waiting for a while, and then I have a question on the chat, and then we'll go to. So, but if people can just raise their hand, it's good, so I can see who's in the queue.
Okay.
Thank you, Amaya, for giving me the chance. Thank you, Nahla, for the presentation, and Sean. I think you get this question probably again. What is the cost per genotype for a breeder like us to run for Blueberry using LGC as a service provider, I guess?
Yes.
Also, maybe the answer depends on the number of samples, but how much did it cost for you to develop these with them? And how much was your invoice when you paid for these 100-something samples that you did?
Okay, it's a little bit complicated. I can give you one answer right now. At this point, as part of the consortium, we have in this year, we have genotyped over 5,000 samples. And for that, we pay around $50 a sample, including DNA extraction. Okay, for both blueberry, for cranberry, $42 a sample. And as I said, that includes DNA extraction, and we get the data back. So we just submit the leaf samples in the plates and we get the data back.
Okay,
and
thank you.
It does depend on the number of samples, but we are continuing I guess the consortium. And if we reach over 10,000 by end of April, the price will go down.
Okay, and then that's for the validated probes, not every probe.
Yes, that's for the 22,000 probes in blueberry and the 17,500 in cranberry.
Okay, thank you.
You're welcome.
Okay, so we have a question in the chat, going back to the previous discussion about, are we differentiating between Oxycoccus and microcarpum when referring to small fruited cranberry?
Yeah. Small fruited is Oxycoccus, and macrocarpon is the large fruited, or the
Yeah, but the question is about microcarpum.
Oh, micro. I don't think. Okay, Jennifer. I don't believe that we had a sample in the testing phase of microcarpon. So I imagine that it would work. It would be a little bit lower recovery, just like we showed in blueberry. It looks like it works well. I mean, in blueberry, the probes were all designed in northern highbush and southern highbush blueberry, but they worked not only in other species in Cyanococcus, which is the section that has the cultivated blueberry, but in other sections. So I imagine that they would work in microcarpon, but I can't say what the recovery rate would be.
So I sort of have a feeling like
Yes.
Go ahead, Jen.
it's a sort of a taxonomic issue. I'm not sure what microcarpon is. Some people I think are calling Oxycoccus microcarpon.
Yeah, I guess
I agree
they
with
split
you. I don't think-
Oxycoccus into two, microcarpon and Oxycoccus, depending on the ploidy. Did I understand correctly?
I don't know.
I don't think microcarpum is an accepted taxonomic determination.
Okay.
I think it's a sort of an old name that's evolved, but I don't believe it's used now. I think it's an old old name.
Okay, but
Microcarpum.
does it refer to Oxycoccus then?
It does. Oxycoccus is the accepted name.
Okay.
As far as I'm concerned, they're the same species.
So then it works in Oxycoccus. Okay.
Good. Okay, so we have Krishna had his hand raised and he wanted to ask a question. Go ahead.
Yes, Krishna.
Yeah, thank you, Dr. Nela. I have one basic question. Like you mentioned non-Cyanococcus blueberries. So what are those? Because I just read that blueberries belong to Cyanococcus genus.
The cultivated blueberry, but there are wild blueberries that are not, and there are other non-blueberry like lingonberry. And did we have lingonberry in there? So I was gonna take you to this slide. It shows the species. So we had angustifolium, that is low bush blueberry. We had Darrowii, which is in the background of southern high bush. We had Eliotii that has been used to introgress those are Cyanococcus. So everything to Tenellum is Cyanococcus species. And then Euliginotum, Ovarum, Padifolium, Vitis idea, Staminium, and Arboreum are non-Cyanococcus. So those are the species that other breeders were interested in, and they wanted us to include and see how well this platform works in these species that are not in the section Cyanococcus. Did that answer your question?
Yes, yeah. Because I was just thinking that all blueberries belong to Cyanococcus, so maybe I was wrong.
No, the cultivated blueberries, the northern high bush and southern high bush, and actually also the low bush. Yeah, the cultivated, yes, does. But there are, others that are not Cyanococcus.
Thank you very much for clarification. Yeah.
Okay. We have one question in the chat from Massimo saying, are you planning to engage possible partners to coordinate a second round of sample submission?
Yes, and that is the plan. That's why I said if you're interested in joining the consortium, please do. And I will be sending information about the next round, after April. The end of April is the first year of genotyping. So after we get all our samples for April, I will send an email to let the community know what to expect for the second year. for 2024.
Perfect.
So yes.
Joshua, you have your hand up. If you want to just unmute yourself and ask the question, go ahead.
Yes. Hi, Noah. It's Josh.
Hi.
Great presentation. One question I have is with regards to the probe development. So I apologize. I was in a meeting prior to this. I didn't get a chance to hear about how the probes themselves were developed. A second part of the question is, you demonstrated that on average you have a SNP every 30 to 50 kb depending on which species we're looking at. Do you think that is sufficient with regards to having a dense enough probe platform with which to carry out and do phenotype genotype associations, or do you feel as though you might need to increase the density of the probe set in order to adequately capture signal when doing various GWAS experiments?
Okay. I'll answer the first question. So it is Rapid Genomics that designed the probes, Joshua. So what we did is we submitted the locus we're interested in, and they designed the probes. And that's proprietary information we don't have access to. The second question regarding whether this is dense enough for GWAS, my understanding is that LD is about 70 kb, and this is way less than 70 kb. So that should be dense enough for GWAS. Did that answer your question?
Yes. I'm just wondering if in the process of, potentially filtering the variants, if someone were to use different filtering approaches, if they might wash out some of that signal or some of the variants that might still be in LD with the trait.
So, as Sean described, you can try different filtering, but, we showed you the results after filtering. We ended up with, I think, 35 kb in blueberry, and how much was it in cranberry? Something comparable in the 30 kb when, half of LD.
That figure of 70 kb as well, I think is a very weak LD. Like if you're looking at SNPs you'd actually filter because of strong LD, that would be in that region of hundreds of kb. I think the 70 kb was an R score of 0.1 or maybe even less.
Okay. Thanks, Sean. Thanks, Nolan.
You're welcome.
Just to add to Josh, to answer that you gave, we do have some preliminary data on multiple GWAS as part of the project. We do get very strong QTL, very good signals. Just
yeah, I didn't mention that because we are using, Josh, we are using them for GWAS, and I haven't seen the results yet.
I figured you probably were. In other crops that may not be sufficiently dense, so that was my initial concern.
Right. As you know, it's crop dependent. So in blueberry and cranberry it is.
Yep. Massimo, since you're on, are you guys applying this in both linkage mapping as well as association mapping populations, or have you just applied it for GWAS purposes?
Well, if you want to go, but otherwise, so we use it for both. We have we use it in both and work in both cases.
Awesome.
Hamid, go ahead.
Oh, okay. So if there's no one else, what was the question I wanted to ask? Is LGC sharing with you the pipeline and the software that you use, or it is proprietary to their company and you don't know what they do with the data? And how confident are you with the SNP calls?
Yeah, Lauren is here. I haven't checked that. We haven't checked that. Lauren is here. She can answer that.
Yeah, so hi everybody, my name's Lauren. I'll be working closely with Nala in the coming year to talk about the next round of partners wanting to use the panel. So I work at Rapid Genomics / LGC as a technical sales representative. So I'll be probably CC'd on plenty of your emails if you have questions about that. So basically, bioinformatics, so the design process can be a little proprietary. The bioinformatics is pretty straightforward. So the SNP calling, we're using, we're writing our own pipelines, but a lot of it is using kind of industry standard for SNP calling, especially for tetraploid calling. So that is not proprietary necessarily how we're going to go through and filter those for you. So each group, as Sean went through the data types, when it comes time to run your samples, we'll ask you if you want your raw data. We'll ask you if you want your VCF file run in tetraploid or diploid mode, and we'll go over kind of the basic filtering steps involved in that, as well as the haplotype files. So yeah, if you have any questions about that, we'll be happy to discuss that with you.
And I mean, maybe based on the second question that you made, maybe about the accuracy of the SNP skull. So far, I guess that between the results that Nana showed about the relationship and the expected relationship and GWAS results that we got so far with this data, there is no sign that there is any concern. So I would say we trust the SNP skull based on the results we have seen so far. So all the relationship that Sean and Nala showed, it was kind of what you would expect, and population structure analysis of some preliminary GWAS that were presented even by, I think, by Patricia at some point. It's indicating that we are getting signals. So
thank you so much. And also we have recently named a blueberry called Rowan. So your last name matches with our blueberry. So
it's meant to
there
be.
you go. Oh, that's my phone. Sorry.
All right, but I don't see any other questions. So what I'm going to do is I'm going to re-share with everybody the link to our YouTube channel. So if you joined it late and you were not able to catch the entire webinar, you can watch it here. It's here in the chat box. And just thank you to Noah and Sean for an excellent presentation, and for everybody. Thank you for participating. And we will be sending you probably a survey after this, which are really valuable for us to just try to make these webinars better and making more accessible to everybody as well. So thank you very much.
Thank you all. Have a great rest of the day. Bye.